So first, my disclosures, I'm affiliated with some different companies. I'm very interested in translation. It's something that is Vice Chair for Research and Translation in BME. I also help other faculty with any potential conflicts of interest are managed by Johns Hopkins. What we find is that those end-capping groups also create the tropism of which type of cells those particles then can be tuned to then uptake and efficient delivery in, even without adding ligands or any targeting groups like that. It's just based on the chemistry. Throughout the polymer, there's then the tertiary amines. That then are able to help with the endosomal escape. So by having a pKa that's in the endosomal pH range, these polymers then have the ability to buffer when protons are pumped in to the endosome, and then they're able to lead to efficient endosomal escape. And then all throughout the polymer structure are ester linkages that then promote quick degradation of the material. And so this is important to release the cargo and also to attenuate any toxicity. So what we find is that these materials are very non-toxic. There's a very high therapeutic window. They can be used 100-fold more compared to other conventional polymeric delivery systems without any toxicity, a wider therapeutic window than with lipid nanoparticles. We've validated these are safe and effective on a range of cells where usually you would need viruses to deliver the genes, and that we're able to do that. We're able to do that non-virally with these particles. So we can have this delivery that cells are usually seen as hard to infect. Maybe you need to use viruses. We can do that non-virally very safely. These materials can be used ex vivo then to deliver your favorite genes, get high yield protein or other kinds of cellular engineering ex vivo. And that we've shown that we can use these in different applications in vivo. So for example, for ocular gene delivery with collaborations with the Wilmer Eye Institute here, we can have a single injection of these particles. We see very good expression throughout the retina in a very robust way. It lasts for at least 8 months. It's very durable. It's very safe. We can deliver these particles systemically to deliver DNA or RNA, including to different types of tissues. So here, for example, is an example of avoiding the liver. You have more delivery to the lung. And then we can also modify the polymer structure to deliver other types of biologics that don't have to be very negatively charged, like proteins. And we can deliver RNPs, or protein nucleic acid together for gene editing. And this is an example in the brain of using that system. And so how we develop them is one of the things we do in our lab here is new types of development of high throughput, high content bioassays to then probe the way these nanomaterials work with cells in vitro. And then we can look, for example, at a library of materials and see how small changes to the structure of the polymer change the delivery properties, such as uptake and endosomal escape. We can look at this in a very quantitative way. So in multi-well plates, we can look at individual cells and see individual endosomal escape events, individual nanoparticle uptake events, and then quantify that. And then use this to see how we can design better materials. And so what we find is that if we move to hard to transfect cells, by engineering the polymer structure, branch structures with lipophilic structures improves the delivery. And that we can see that, for example, increasing the branching of the polymer increases the endosomal disruption, and this leads to higher transfection. And so by taking these learnings, we can then home in on which types of materials work best for certain functions. So, for example, here's showing delivery with these materials of siRNA, mRNA, or plasmid DNA, and then quantifying the levels of nanoparticle uptake, the levels of endosomal disruption, and then ultimately the levels of transfection. And then what we find is with increased hydrophobicity, for example, by making the side chain that's hydrophobic a larger component, or increasing the tail length of a hydrophobic side chain, we can get this improved transfection that compared to leading commercially available reagents like polyethylamine or lipofectamine 3000 is very superior for transfection because it's especially better at endosomal escape. And so we have about an order of magnitude higher endosomal escape than leading lipid-based and polymer-based nanoparticles. And that's a big barrier for intracellular delivery. So using this approach, we can then find materials that are good for certain applications. And so one of the things we look at first is for intramuscular injection of a vaccine, and could we boost the efficacy of that? And so this is what the specific polymer looks like. We blended this hydrophobic and hydrophilic side chain. We blended in a little bit of lipid PEG. The particles are about 100 nanometers in size. And using our library approach, we could then find the specific structures, like specific end groups, that then lead to higher transfection. And this wasn't known a priori how very small changes to the structure, just the very ends of a polymer, the whole middle of the polymer is the same, make such a dramatic difference to the transfection ability. When we look at the intramuscular administration, this is something that works well, usually with nucleic acids in a mouse model to do a high volume injection right in the muscle. But we found with the polymers, we're able to boost that transfection further. This is a log scale here, luminescence from the mRNA, and to find polymers that work really well for ion administration. And so this is the structure of the polymer. And we found that this would work very well at even low doses. So this is doses of just 100 nanograms of nucleic acid. So it's a very low dose, and yet we see, in this case, neutralizing titers of, this is for a vaccine against rabies, so that was able to work well. When we, a question could come up, well, if they're degradable in water so easily, how does that work with different kinds of collaborations? And what we found is we can actually freeze them, or we can lyophilize them, and they'll stay stable long term, and then we can just reconstitute it with water and administer it. And so 2 years, it can be stored at -20, works as well as fresh. And it lyophilized even works pretty well in the fridge, just at 4 degrees for several months. So while this could be useful for generating antibodies for a really potent genetic vaccine, we really want to get a strong cellular response to. And so we could do that if we could target antigen-presenting cells well. And antigen-presenting cells are like the director of the immune orchestra. So if we get the right signals to them, they can then mount all the different T-cell responses we're looking for. And so leveraging this chemical library then to different polymer structures, we're able then to change where we see expression. And so with the small chemical changes, no ligands are needed. We can then have expression in different areas of the animal. So there's some polymers, like this PD, that go right to the liver, and we see a lot of expression in the liver. This is what you see with lipid nanoparticles. But then we have other ones that will avoid the liver and instead have more expression in the lungs or in the spleen or all over the animal that can then be useful for other gene therapy applications. And in this case, we're very interested in the spleen, and we're interested then in the cells in the spleen we can target, which we found were dendritic cells. So we could administer this IV and see the expression be predominantly in dendritic cells, which is just what we were looking for. So then we designed polymers that were very good at doing this. So we would have a formulation we can mix in other nucleic acids with the mRNA. The mRNA encodes an antigen. It would be expressed in the tumor. And then we could add immunostimulatory nucleic acids like CpG or poly-IC, make that particle, inject it systemically, goes to the spleen, transfects APCs. Those APCs will have a peptide encoded from the mRNA presented in their MHC on the surface to stimulate the right CD8 T cell that becomes activated, proliferates, and then could go after the tumor. And so that's what we see. We initially looked in vitro at dendritic cells and DZ2.4s and then primary dendritic cells, and we found that with these formulations, especially increasing lipophilicity of that side chain, we got very high rates of transfection. And then we found that this translated to in vivo work as well, that we were able to see very good transfection in the spleen. We could blend in these immunostimulatory nucleic acids and then see activation as well. And so this was about 5% of all the dendritic cells in the spleen were transfected. So then we wanted to move to the functional models. And so we looked at B16 ova first. And so that has an antigen on the surface, and we could have mRNA that encodes the relevant antigen. And what we found is that particles that had that mRNA for ova, as well as CpG or poly-IC that were just mixed into the particle, were able to significantly reduce tumor growth and enhance survival. And then when we looked at the blood in those animals, 2 weeks out, we saw that almost half of all the CD8 T cells were the CD8 T cells that would recognize that antigen that was encoded in the mRNA. So this is a greater than 10,000-fold expansion of those T cells, but it all happened in situ. So instead of taking T cells out of the body, ex vivo engineering them and putting them in, it was just an injection of the particles, and all that cellular engineering happened in situ, in vivo, in the animal. Then we had this 50% response. And then we looked at other models. So with B16F10, where you don't have ova, this is more difficult, but we used mRNA encoding TRP2 and GP100, and we did see this extension survival as well. And then we moved to another model that was more immunosensitive, MC38, which is colorectal cancer. And then here the results were even more striking, with having very strong reduction of the tumor growth, the purple line for the combination. And then half the animals actually, their tumors fully regressed away, so that we administered these particles, and there were 2 injections of nanoparticles and 1 injection of anti-PD-1. And we saw half of the animals, the tumor shrank away. And then they survived re-challenge. So when they got new tumors on the opposite flank, 2 months later, no new therapy, the tumor shrank away in all those survivors, that they had this long-term durable resistance to those tumors. And then we looked at the T cells, again, we saw that in this case, more than half of them were specific against the antigen. So that can work well for new types of genetic vaccines that mount a cellular response in addition to just thinking about the antibody response. But what about if we don't know the antigens ahead of time a priori? And so for this, what we want to look for is a personalized response to a tumor without having to know anything about the tumor. And then it's an off-the-shelf particle, lyophilized particle that can just be reconstituted, injected in the tumor to then create this very strong immune response. No biopsy or sequencing required. And it's something that can be inexpensive to manufacture and widely accessible. And so to do this, we're looking back at the antigen-presenting cell. But instead of doing delivery of the antigen-presenting cell this time, we want to mimic what an APC does. And so it has a signal 1, which gives the specificity in this antigen presentation, signal 2 for co-stimulation, and then signal 3, which is a secreted immunostimulatory molecule. And these 3 signals come together, and that's what helps the T cell know to get activated. And so we want to recreate that. And so our strategy is we look at a tumor, and the tumor has its antigen presentation, like that's the part it has. But by adding our nanoparticles to it, the idea is, could we force expression of the co-stimulatory molecule signal 2, the secreted immunostimulatory molecules, so that for a T cell, this new tumor-derived APC can now look like, smell like, taste like an APC, and then the T cell would be able to be activated and could proliferate and go after whatever antigens are on that tumor. Again, we don't have to know it a priori, and it could even be many different antigens. And so we're sort of flipping the tumor from being a bad guy to being a good guy that shows a wanted poster to really teach the T cells how to recognize it and then its other brother cancer cells throughout the body. So that was the strategy. And so first we used the same assays as before to identify polymers that could be very good at transfection of cancer and also very specific of cancer, that they could transfect just the cancer cells shown in red here, and then the transfection makes them green without transfecting the healthy cells. This is healthy hepatocytes, for example, and liver cancer, and the nanoparticles just deliver to the cancer cells. And so identifying the polymer from the library that does that the best, we then took this in vivo in a model for B16F10 melanoma. And what we found is that this reprogramming actually worked really well at reducing tumor growth. And then we got long-term survivors with this approach by having 4-1BPL as a signal 2, IL-12 as a signal 3, and in the background of anti-PD-1, we had long-term survivors where anti-PD-1 alone led to all the animals dying within a few weeks. What these animals look like is where the tumors used to be, there's now white patches of fur with these long-term survivors. And then if we gave them tumors on the opposite flank, no new therapy, they had resistance to those tumors. They eventually did succumb to the disease in this case, but they lived longer. And then we looked at another model, and a lot of this work is led by a talented assistant research professor, Stephanie Zhang, being me. And we looked at C38 colorectal cancer, we saw much more striking results where in this case, all of the tumor shrank away following this treatment. And then there was 100% survivors. And then when they were re-challenged, 100% of the survivors, the tumor shrank away and they continued to be survivors, where age-matched controls, they got that re-challenge, died just like the original cohort died with this reprogramming approach. And so where we're looking now is things like for metastatic disease. So if we have melanoma in the lungs, as well as in the flank, if we treat just the flank, what happens to the lungs? So the flank will shrink down, but then if we look at the lungs, with control particles and anti-PD-1, there's still lots of tumor cells in the lungs. The tumors are luciferase positive here. But when we add our particles, we see that the tumors in the lungs all shrink away. There's no treatment to the lungs. The treatment is just to the tumor that's in the flank. But what happens is that cellular immune response will go through the body and look for wherever other cancer cells are. And if they find them in the lungs, those CD8 T cells attack them in the lungs, and then the lungs come out very healthy. And when we look at the area around the tumor or around the lungs, we see CD8 cells infiltrating in. So I hope I've shown how biodegradable polymeric particles can be useful to program the immune system. Chemical properties of the polymer were very important, like the end group and side chain to get the specificity and the efficacy. These approaches can work with targeting without having to have ligands, or in some cases, without even having to have any tumor sequencing or knowledge of the tumor a priori, that still have this personalized cellular immune response. And nanotechnology like this opens up new opportunities for immunotherapy for cancer, but also for autoimmune diseases, tissue transplantation, infectious disease, and other types of applications. I'd like to just give a quick shout out that there's a new center at Hopkins called the Johns Hopkins Translational Immunoengineering Center. It's the first NIH-sponsored immunoengineering center in the country. You can learn more on this website. There's winter workshops available, and the technologies that we're creating here are available for other researchers here, at Hopkins and around the world to use if it's helpful for them in their research, like this delivery technology. And so with that, thank you so much for your attention. I'd be happy to answer any questions.